Okay, good afternoon. Thank you all for participating in today's media availability. I'm joined by Dr. Don Yealy, UPMC's Senior Medical Director and the Chair of Emergency Medicine, Dr. Oster Mariquin, UPMC's Chief Healthcare Data and Analytics Officer, and Dr. Derek Angus, UPMC's Chief Healthcare Innovation Officer and Chair of Critical Care Medicine at the University of Pittsburgh. They will update you on our clinical data and experience with COVID-19 and show what has changed over time and how that continues to inform our actions. And they will also discuss our ongoing adaptive clinical trials and how they are helping to inform us and to take better care of our patients. After they offer their remarks, we'll take your questions. Dr. Yealy.
Thank you very much, Paul, and thank you all for showing ongoing interest in the pandemic and the UPMC experience with COVID-19. As we've been saying for many months, COVID-19 came to all the communities that we serve here at UPMC, and it's not going away. Everyone, that's all of us in our communities, including the medical professionals, is learning how and adapting to living with this virus, something that we will continue to do for many months. As you'll soon hear from Dr. Mariquin and Dr. Angus at UPMC, we are learning while we deliver care to you and to your loved ones. And we're learning fast, which is helping to improve our outcomes for all of our patients. Key measures, including the UPMC COVID-19 test positivity rates, the amount of time our patients spend in the hospital, their need for intensive care or the use of ventilators, and those are machines that help you breathe, and the death rates are all far lower now than they were earlier in the pandemic. That's very good news for all the communities we serve in Pennsylvania, in New York, and in Maryland. It isn't a victory declaration, though. It shows that we can impact this infection with our actions, and that includes not only you in the public, but healthcare providers. Why are we seeing these better results today compared to what we saw in March and April? Well, first, we have better treatments, including steroids, and more effective clinical protocols. And we know better how to use all of the care steps for patient care, including positioning and breathing support. We are seeing from our own data, as well as data from other locations, that suggestions exist that the virus is changing in important ways. People are now experiencing less frequent bouts of severe virus impact. Perhaps that means that the virus is less virulent than we once thought or experienced. That doesn't mean that we can all go back to normal as we knew it before COVID-19. Wearing a mask as much as possible, keeping distance whenever possible, staying home when you're sick, and washing your hands frequently remain critical to keeping this virus in check so that we can reopen our businesses and our schools as safely as possible. We thank our dedicated staff and, more importantly, all the communities that we live in and we serve for taking these steps. You followed public health guidance, and you demonstrated that we can care about ourselves and about each other. We can't predict what the future holds for this virus in terms of its spread and its severity, and especially as we enter the flu season and we spend more time together indoors. What was key, what is key, and what will continue to be key is clear. Wear a mask whenever you're near others, and wear it correctly. Cover your nose. Secondly, make sure that we all protect those who are vulnerable. This virus looks for the most vulnerable. Keep them safe and be safe around people who are vulnerable. That includes the elderly and those who have conditions that affect immunity, their heart or their lung functioning. These actions will help us sustain and improve our collective COVID-19 experience, and if we all do these actions, we can avoid impacts of broader closures of our workplaces, businesses, schools, and other activities. At UPMC, we remain committed and vigilant to be prepared to care for all of our patients and our staff should the virus surge again. We will do all that is necessary to make sure that we can treat you no matter what your healthcare need is, and we can do it safely. we will also create the new knowledge and the new care processes that will help now and will help moving forward. You expect this from a world-class medical center, and we'll deliver it. Excellence in care and in innovation. That excellence includes approaches to testing. While many push for expanded use of new rapid tests for the virus, we use an approach that is based on the facts and the science. We need fast and reliable tests to contain this virus, just as we need to develop safe and reliable vaccines. And we will fully evaluate any new rapid tests to make sure they meet both of those criteria before adopting them. Now Dr. Mariquin will explain in more detail what our clinical data are telling us about what has and hasn't changed since the pandemic began.
Thank you, Dr. Yili. As Dr. Yili explained, the clinical data we have tracked and analyzed closely throughout COVID-19 are showing us some important lessons about how to better manage this illness. Let's start with testing. While we all wish that we could do more, we are in fact testing 2.5 times more COVID patients per day in the last 2 months compared to the prior period. And the test positivity rate is only slightly higher at 5.28% currently compared to the spring, which was 4.94%, suggesting that the people we serve are mostly taking the right precautions to protect themselves. While our testing has grown dramatically, our number of hospitalized patients has not. The average daily number of patients hospitalized with COVID-19 since July is approximately 2.8 times higher than in the spring. But daily discharges are 3 times higher. Our discharges are outpacing our hospitalization rate. Patients are getting better, and they're getting better faster, at least a day faster, as a matter of fact, than before. The best news of all is that in-hospital mortality rates are trending down. Surprisingly, the patients whom we admit are not much different than those we treated earlier in the pandemic. That is, current patients are similar in age and have similar comorbidities like diabetes compared to those hospitalized in the past. So why are the outcomes better in the past 2 months? We think part of the answer lies in the way that we have changed our care for these patients as we've learned more about this previously unknown disease, as Dr. Yili noted earlier. For instance, our data show that more than half of patients since the end of June have received dexamethasone or other steroids, while the use of remdesivir has declined. And we are more judiciously using mechanical ventilation. These are just some of the factors at play in this incredibly dynamic disease. We continue to gather data and refine our analysis so that we can continue improving care and outcomes for our patients. Dr. Angus will tell you more about the lessons we have learned, particularly through our leadership in rigorous and adaptive clinical trials.
Thanks very much. So as Dr. Mariquin just told you, our data show that we're learning how to do things better over time, and maybe also the disease itself is changing. Specifically, what I'd like to talk to you about today is some positive news from our, sorry, are you having trouble hearing me? I'd like to report some positive news from our global Remap trial. A commonly available, inexpensive class of drugs called corticosteroids, we have found considerably reduces the odds of adverse outcome, death and or duration on life support in critically ill COVID-19 patients. These results, coupled with those of other global corticosteroid trials, were published yesterday in JAMA, or the Journal of the American Medical Association, and in fact also led yesterday to the WHO to issue new updated guidelines in this regard. And it's important to note we were delighted not only to be involved in the leadership of the Remap trial, but to have UPMC actively participating, and we had a significant number of patients from UPMC hospitals participating in this trial. I spoke once before about Remap. As a reminder, Remap stands for a randomized, embedded, multifactorial adaptive platform, which is a new kind of way of thinking about doing randomized trials. The gold standard for testing whether a treatment actually works has for a long time been the so-called randomized clinical trial, or RCT. But the way in which we've done RCTs has traditionally been to look at one treatment at one dose at a time. Every patient is randomized 50-50. That's sometimes something that someone doesn't necessarily want to participate in, and furthermore, it can take months or years to get to an answer. What we want to do is think about trying to generate just as robust information, but do it more quickly and do it in a way where patients have a greater chance of benefiting. So Remap is designed to simultaneously test multiple combinations of potential therapies at the same time, and this increases the efficiency and the speed with which we generate information, which is always valuable, but particularly valuable during a pandemic. And so, for example, within our current Remap CAP COVID program, we've actually been testing multiple different treatment regimens, including various doses and combinations of even simple things like vitamin C, but also convalescent plasma, blood thinners, antivirals, and other drugs. The other thing that I just want to remind you all, we were involved in the design of Remap, and we've been very vested in delivering it here at UPMC, but it is actually a global initiative. In the corticosteroid domain that I was alluding to, we actually enrolled patients at 121 hospitals at 8 countries around the world. And it's also crucial to getting to information quickly. So coming back to the actual corticosteroid, many of you have probably heard about dexamethasone. This is one type of corticosteroid. And there was a trial published from the United Kingdom earlier this summer that suggested that there would be benefit with dexamethasone. What was going on in Remap, we were actually testing another steroid, hydrocortisone. It was being given in a very similar dose and in similar patients, critically ill patients. It turned out that with hydrocortisone within our trial, there was a 93% probability that hydrocortisone was superior to not giving hydrocortisone. At the same time, we had colleagues conducting traditional randomized trials in Brazil and France, and actually all of these studies came together, and they were all published yesterday in JAMA. And so after the initial good news from June on dexamethasone, what we did yesterday was really help provide definitive information, and that's in part why the WHO has now issued this very clear guideline on giving corticosteroids, hydrocortisone or dexamethasone, to critically ill patients. This is only possible because of a global community of physician scientists coordinating across different languages, different countries, working together and sharing data at a speed and a level that I think has been unprecedented in order to generate definitive answers. And I feel incredibly proud that UPMC and the University of Pittsburgh was absolutely at the center of that initiative. Now, I do understand, as I already alluded to, that randomized trials can often feel slow and they take too long to generate information. I think, as we've shown with Remap, that does not need to be the case. We have absolutely been able to generate robust, actionable information at the speed required to respond efficiently in this epidemic. This is part of what we would say is learning while doing. It allows us to arrive quickly at clear guidance on how to treat patients. I would also say it's a way of thinking about how we can have a structure where we're trying to learn from every patient we're treating. So that, for example, in this trial, when someone in Amsterdam agrees to be enrolled in the trial, they produce data that can then help us treat someone in Altoona. This pandemic has shown more clearly than ever why this approach is necessary to achieve speed and to generate effective medicine. Thanks.
Thanks very much, Dr. Marroquin and Dr. Angus. What you say supports what I see daily at UPMC, and that is our COVID-19 patients are doing far better now than at the start of this pandemic. We have a team of gifted physicians and dedicated nurses and other healthcare experts that are doing the things that matter during these trying times. We will use our excellence and we'll leverage all the knowledge that we have and that we find in other locations and the abilities from around the world to help eradicate COVID-19 in all the communities that we serve. UPMC remains ready to care for you and your families throughout this pandemic and beyond, and we'll keep you healthy.
Thank you, doctors. And now we'll take questions from the media, all of whom are joining us remotely. To ask a question, you'll have to go to the participants icon at the bottom of the page and press the Raise Your Hand button. That will put you in our queue. I will read your name and your line will be unmuted for you to ask your question. Please remember to unmute the line on your end as well. We will try to call on as many different media outlets as we can in the time that we have allotted. We do have a few email questions, and I'd like to turn first to Dr. Angus. What are your thoughts on the FDA's emergency use authorization for convalescent plasma and vaccines?
Thanks, Paul. So let me just so that everyone's clear. So convalescent plasma is when one takes from the blood of patients who've recovered from COVID-19, one wants to draw the plasma that contains antibodies that person mounted in order to overcome SARS-CoV-2. And so then the issue is, if we give this plasma to someone else, then we can transfer the antibodies in and we can help that person recover. So convalescent plasma has felt like a good idea for a long time. In fact, there are reports of trying to give convalescent plasma in the 1918 Spanish flu epidemic. The problem is, there have never really been robust randomized clinical trials that actually demonstrate benefit. And so it continues to feel like a good idea. There's strong rationale for it, but we're not actually sure of the benefit. I fully understand the pressure the FDA is under because we can now harvest and make convalescent plasma available. We've actually given convalescent plasma around the United States to around 70,000 individuals. But disappointingly, it's never really been randomized. Actually, here at UPMC, we were attempting to randomize to early versus delayed, and elsewhere in the Remap program overseas, we are effectively randomized. But the problem about making the EUA available here is that that's actually stopped any immediate attempt to get a randomized trial up and running in the United States. And so now it's going to be weeks at a minimum before we can even start to begin trials. So I would say that the EUA feels like a response to a compelling need, but it's probably only going to slow or delay the speed with which we get to really understand whether convalescent plasma has an effective role to play.
We think that there's great opportunity to improve how it is we bring new therapies, even if they're new therapies behind an old idea, how we do that more efficiently. And an emergency use authorization is one way to do it, but it may not be the most effective way to make sure that eventually we're using the best possible therapies. We want to get better at this. We do not want to depend on a laborious, slow process in a pandemic that requires quick action. We need to be thoughtful about it so that we can continually respond and learn and eventually get better, not just respond quickly and not gain as much information as we might be able to.
Okay. Thank you. Let's go to Paul Goff, Pittsburgh Business Times. Paul, go ahead, please.
Hi, can you hear me okay?
Yes, go ahead, Paul.
Thank you very much for the time. Wondering if you can tell me what UPMC is doing to prepare for the rollout of a vaccine, either one that's being developed at UPMC or will be through CDC, what role the health system has.
So UPMC and the University of Pittsburgh continue their efforts on 3 distinct different vaccine approaches. Those are still ongoing. They're not at human phases yet. You've heard about those in previous press conferences. Secondly, UPMC and the University of Pittsburgh are enrolling and participating in the Moderna vaccination trial that is happening not only in the country but across the globe. We want to be a part of evaluating any very good idea about vaccination. What's important to think about is that vaccines will potentially become a valuable tool in the response to COVID-19. By themselves, they won't likely be the singular changing action or solving issue for COVID-19. They'll be an important part of any response. And you might say, why is that? Not everybody mounts a good response to a vaccination. We'll learn more about it as we participate in these trials. And some folks actually decline the opportunity, and we have to work on that. So there'll be lots of reasons that we can improve on vaccination trials and hope for them to be a part of the solution, but I don't think they'll be the complete solution for COVID-19.
Another email question. We'll get to the media in a minute, but please, again, for those members of the media, please press the raise your hand button, and that will put you in our queue. We'll unmute you, and you need to unmute from your end. So, or we're waiting for that. We'll get to another email question. I think this goes to all three. What should people take away from the data that Oscar presented in terms of the severity of the illness? You've said perhaps we're dealing with a lesser strain.
I mean, I would say that what we have seen with this disease is that the outcomes that we are seeing are the result of multiple factors. Certainly, there are issues related to the virus and its pathogenicity. There are issues related to the host that the virus infects. And there are also issues related to how we care for our patients. And then lastly, there are issues related to how we as a society behave to try to mitigate the spread and contagion of the virus. And we have certainly seen significant changes in how we take care of patients, for how we have learned, as well as the way that our community has adhered to the recommendations for masking and other things, both of which we believe are among the things that our data show that have had an impact in the improved outcomes that we see in our patients.
Yeah, it's impossible to separate what we do as humans from what the virus does. It depends on us for propagation and for illness. And so the changes that we see are not isolated to what happens in UPMC or any of the communities that we serve. It's been noted around other parts of the country, including New York, that the pattern of illness has changed and appears less severe. In part, that may be due to virus changes. It has a lot to do with what we choose individually to do and how we adhere, has to do with public health interventions, has to do with the tools that we have available to treat. They all play together and there's an interaction. What's undeniable is that there is a change in the clinical patterns. We're not seeing as much severe illness. We still do see severe illness in some, and it has a tendency to be the same types as we did before. Vulnerable people, those who are older or have serious underlying conditions. We need to stay focused on this. The virus changing will not by itself get us away from those important activities as community members or as healthcare providers.
Okay. Let's go to Gabriella DeLuca at WPXI TV. Gabriella, please go ahead.
Hi. So this question is for all of the above there. I guess the CDC has told states to prepare for a vaccine by the beginning of November. Do UPMC health experts think this is actually possible? And do you feel that if a vaccine is available, would it have all sufficient testing to prove safe?
So I think we would share the comments by others in the national media that we have watched an unprecedented speed of vaccine development. I don't think we've ever faced a global threat like this that has seen such a wonderful mobilization of both pharma companies and academic institutions to get us to this point. Having said that, we're now at the point where we're launching these phase 3 trials, and Dr. Yealy just talked about how we here at UPMC and Pitt are participating in some of these trials, and we absolutely would want as many people in the communities that we serve to sign up for those trials. These phase 3 trials are testing vaccines that have made it through earlier hurdles. There's some reason to believe that they look pretty safe. You know, they're well designed, but they don't yet have adequate proof of efficacy. That's the point of the trial. Once these trials begin, the clock is really a function of just how fast we can get people enrolled, and then we just have to wait. I think we and many would say that it is extremely optimistic to imagine that we'll have any kind of truly robust signal by November. Having said that, everything about vaccine development has proceeded at a faster pace than previously. I don't think we would disagree that it's good to be ready so that on the day that a vaccine is ready to roll out, that we're ready to roll it out, because it won't work unless people take it. Having said that, we would totally say that we only want to roll out a vaccine that has been shown to be adequately safe and efficacious.
We want a safe and effective vaccine, and I think we can do it quicker than we've done in the past, and we want to be a part of that process here at UPMC. We won't use something that isn't both protective from the infection and keeps you safe. We think that there are opportunities to do this much quicker, and getting ready in advance is an excellent idea. But we, together with others, want to carefully weigh those data when they're available and make sure that we're doing the right thing.
Okay. Let's go to Bill Kibler at the Altoona Mirror. Bill, go ahead.
Thank you. Can you hear me?
We can, Bill. Go ahead.
Okay, thanks. There was a New York Times article recently that suggested that the way PCR testing is done makes those tests so sensitive that most of the people it registers as positive for COVID-19 are carrying so little virus that they're not contagious, and therefore wouldn't really need to isolate or have their contacts traced. I was wondering if one or all of you could comment on that.
Well, we're familiar with the New York Times report and the science that it's based on. And what a PCR test does, to make sure everyone understands, is we take a specimen from usually the back of your nose. There are some other locations that don't work quite as well, but generally from the back of your nose. And we extract out the genetic material from the virus separate from any of your genetic material. And then we often have to amplify it. In other words, bring it up such that it can be measured. And what you're talking about is sometimes you have to multiply whatever the sample is enough to be able to detect very small amounts of virus. And the question comes down to, what does that mean? If I have to amplify something many, many times, is it really an event? And what I'd say is that we're learning more and more about the disease. First and foremost, it tells us that you've been in contact with COVID-19. The amount of virus in the back of your nose and how it interfaces with how much illness you have that day, how much you might have in a few days, and whether or not you can infect someone, really are three different variables that not only are we learning a lot about, we don't fully understand yet. In general, people who have less virus are less infectious to other people. We don't know those thresholds, and remember, it's the person on the other side that has just as much to do with the interaction. If you're vulnerable, you're more likely to either acquire the virus or have a bad outcome from it. That's why you see these observations about, should we be worried about people with less symptoms or less amount of virus or not. It's key to make sure that we focus on not only those who carry the virus, but those who might come in contact with it, and protect the latter as much as possible. You protect that by wearing a mask yourself if you're carrying it, even if you don't know that you're carrying it, and wearing it when you want to try to ward off. It protects in both directions.
Okay. Well, let's go to Shelly Stahlsmith at the York Daily Record. Shelly, please go ahead.
Hi, can you hear me?
We can. Go ahead, Shelly.
Okay, thank you. Last night at a school board meeting, Dr. Wayne Sebastianelli, the director of athletic medicine at Penn State, said that roughly 30 to 35% of the Big Ten athletes who tested positive for COVID now have some signs of myocarditis, however you pronounce that. Is that something that high school parents should be worried about with their kids, or is this just the really elite athletes who you're seeing this in?
Well, the answer is that we know that COVID-19 affects much more than just the lungs. And as a matter of fact, there are some of us that have acknowledged that it is a vascular disease at its core as well. And there's been reports of myocardial involvement when the virus infects patients. And I would say that that is not something that is restricted to just elite athletes. I mean, what we see with elite athletes is that they're getting tested more often. And as part of the process of their testing for being elite athletes is that they also get testing for cardiovascular conditions and whatnot. But it should be certainly assumed that the virus does not discriminate in that sense, whether somebody is an athlete or not, and the likelihood that it can affect other organs besides the lungs. And in this case, the myocardium, which is the heart muscle, is certainly something that has been described clinically, and that would be something that could happen to folks that get infected, regardless of whether they are athletes, elite, or not.
I think the bigger question is going to be, does this matter next week, next month, or next year? Think of it, we are 8 months in the United States with our experience in COVID-19, and because we can detect some heart changes in the middle of an infection, doesn't actually give us as much insight as we might hope for into predicting how that person will be months from now, years from now. And those are the data that we still need to collect. So simply put is, I'm not surprised in a group that gets highly surveilled that you find evidence that the virus isn't just in their lungs, that it can affect other organs. That doesn't surprise us. What it really means is still uncertain right now.
And I may just add that it's not just that COVID-19 is the only virus that can affect the heart, right? There are many viruses that can have an impact on the heart, and there are specific conditions that we call viral cardiomyopathies that we treat. And so it's something to be aware that in this case it's not a novelty specific to this virus, but as we are learning more of the disease, as Dr. Yili explained, we are starting to see that it can affect many different organs, including the heart. Now, the long-term implications of that are still unknown because, you know, these patients, we have to follow them to see what are the clinical manifestations of those findings.
Okay, thank you. Let's go to Bob Mayo at WTAE TV. Bob, please go ahead.
Hi. Yes, just to explore a little bit more the observation once again that the virus may be changing. And can you clarify and elaborate, is that entirely based on observations of effect and trying to infer from that? Or is there any way, or has anyone done anything to examine the virus itself and say, there is on a DNA level or whatever, a documented change that could be associated with that? Or is it entirely based on observation and inferred from observation of what you're seeing?
So the initial evidence came primarily from our clinical observations, and those are our observations across all of UPMC, but also in many other locations, in New York, in Europe, in Asia. People have noticed the same basic patterns. So what we know is that the clinical patterns have changed. Others have looked at the virus itself and seen changes that affect how the virus can not only enter a body, but what it does after it enters a body. Those things aren't conclusive yet, but when you put them together with the clinical observations, you begin to realize that one possibility, and I'm not here to declare it as the final answer, is that the virus is changing. We've known for centuries that viruses change as they interact with their hosts, and in that case, we're the host. None of this is terribly new. I can't predict for you what that will mean in the future. It by itself is not the solution. So the short answer is we began with clinical observations, but there are now data from essentially cellular levels that confirm the virus is changing. What that means is still yet to be told.
Okay, next question. Maria Simbra, KDKA TV. Maria, please go ahead.
Hello. Hi, a couple questions. I have two questions for Dr. Angus. First, about the hydrocortisone. In the JAMA article itself, it states, no treatment strategy met pre-specified criteria for statistical superiority, precluding definitive conclusions. So you're referring to a superior probability of better outcomes, but not outright statistical superiority. And then my second question has to do with dexamethasone. That study was in more than 2,000 patients with death as an endpoint. What now with dexamethasone? What becomes of it?
So these are quite linked questions. So a couple of things. The dexamethasone study essentially was announced to the world on June 17th. There were several other research groups around the world who were also conducting randomized trials in corticosteroids. And essentially on the day that that result was published, in part because it was associated with an improvement in mortality, although only in the sicker patients. It's important to remember in the recovery trial, general hospitalized patients who were not requiring excessive oxygenation or ventilatory support, there was no benefit in that group. But for the sicker patients, the result was seen as so compelling. Those who were doing randomized trials around the world essentially overnight felt like clinicians at the bedside enrolling patients would feel uncomfortable with further randomization. And so Remap CAP stopped randomizing to steroids on that day and moved rapidly into the analysis phase. And so did our partners in France, our partners in Brazil. There was a study running in China and so on. We also then got together with the WHO and agreed not only to analyze all our data individually, but to pool all the data together. So what was published yesterday in JAMA were the individual reports of the three biggest trials, Remap CAP, the Brazilian trial, and the French trial, together with a meta-analysis that we participated in that had all of these data, plus data from other studies, slightly smaller studies that have not actually been published yet, but all the data was put together. So in the Remap CAP study, had the rest of the world not been doing any studies, then at 93%, we would have carried on randomizing. Our internal statistical trigger was set at 99%. So if we were the only one studying steroids, we would have kept going, and presumably in a few more weeks, we would have bumped from 93% probability up to 99% probability. But we're part of a global community. And so in the accompanying paper, the meta-analysis, you can see what happens when you put the findings from our study together with the other studies. And there's a couple of key points. First of all, it looked like it didn't matter which steroid, whether it was dexamethasone or hydrocortisone, or maybe even methylprednisone, they were all pushing results in the same direction. And so there was a strong overall signal across the joint trials. That was that odds ratio of 0.66. So only 2/3 the odds of dying. That's a significant benefit with both giving dexamethasone or with giving hydrocortisone. And so the conclusion, which is reflected in the WHO guidelines, is that treatment with either dexamethasone or hydrocortisone should absolutely be given to any patient who is critically ill, requiring a lot of oxygen support and so on. I hope that's tried to answer both questions together for you.
I think it's important to point out, and Dr. Angus is being modest about this, this is an example of a new approach into bringing scientific knowledge to care. Instead of multiple different groups competing and continuing in a silo by themselves, they actually came together when the first signal of benefit in the same basic class of drug was seen, and did the right thing, putting information together instead of just continuing to enroll more people to make sure their personal trial got finished. I think this is the type of responsiveness you not only expect from us locally and regionally, but what you'd want out of caring, intelligent, and committed scientists across the globe.
And I think for patients who sometimes might hesitate as to whether they should participate in a clinical trial, the fact that these are being carried on by ethical professionals who really have put their careers to do this in the best way possible to gain evidence, but not at the risk of delaying the treatment that patients might benefit, should be seen as a tremendous sort of victory as to how the medical community is balancing the opportunity to derive insights and evidence, but at the same time providing the best therapy for our patients.
A related question was emailed to us concerning remdesivir. Isn't this a treatment that has proven successful against COVID-19?
So let me walk back again to the steroids question. What filled me with trepidation after the recovery trial was that the other trials might not align with recovery. Steroids have been around for a long time. And indeed, Tony Fauci wrote the editorial in the New England about that British recovery trial saying, it's a lovely trial, but it's just one trial. We would like to see the information as consistent across trials. And so what was so clear about the steroids was the consistency of the signal across all the different trials. In contrast, it's been a slightly rockier road for remdesivir. There was an initial report that was published in the New England Journal of Medicine for a study conducted by the NIH, which was encouraging in that it cut the speed with which people recovered from 15 days to 11 days with a 10-day course in comparison to placebo. But it was there was no statistically significant reduction in mortality. And then just about a week or so ago, a second study funded by Gilead actually was published in JAMA. And I wrote an editorial on that with Erin McCreary, who's a pharmacist here at UPMC and helps run our antimicrobial stewardship program. And when we looked at that study, the answer was complicated. They compared no treatment to a 5-day course or to a 10-day course. And the 5-day course looked like it was maybe better, but the 10-day course looked like it wasn't better. And so it felt a little bit like 1 step forward, 2 steps back, or 2 steps forward, 1 step back. I think there's lots of reasons to think that remdesivir could be effective, but remdesivir is quite expensive. And we do not have as clear a signal at this point. So our position on remdesivir actually would be, it would be nice to see larger definitive trials on remdesivir to understand exactly which patients should get it at what dose, and in particular, what's the role of remdesivir in patients who also get steroids.
It's important to remember that some patients might benefit from either one of these drugs. They work completely differently. Remdesivir affects the virus. The steroids affect your response to the virus. Some people might benefit from both or just one or the other. And that's why it's hard when you're trying to keep up with this through the media, trying to understand which one should I have. Why don't I just take everything early? The answer is they don't work that way. And it has to be a careful decision based on the data and where each individual is at in the illness.
Okay, let's go to Jason Mast at Endpoints News. Jason, please go ahead.
Yeah, thank you so much for doing this. Basically, two questions here, somewhat related. One, I'm familiar with some of the concerns about getting patients to enroll in an RCT following this EUA, but I'm curious about why there would be a delay in starting a trial or expanding this Remap cap trial of plasma in the US because of the EUA. And then secondarily, I was curious sort of about maybe why Remap and sort of this approach wasn't more widely used in the US, similar to what went on in the UK, and if there was any interest from other hospitals, other governments, or the government.
Sure. So that was I'm forgetting the first part of the question again.
Why would it be?
Oh, yes. So an EUA is actually a complicated approval from the FDA. Before the EUA, there was what was called an IND, an investigational new device or new drug, an approval under which research could be conducted. And indeed, the Mayo Clinic held an IND that was allowing those 50, 60, 70,000 patients to get convalescent plasma around the United States. When the EUA was issued, that IND closed. If the EUA had actually been a full approval, then the FDA would be saying you can give convalescent plasma for COVID-19. And if that were the case, if you wanted to do a trial, you wouldn't need an IND because it would be under the approved label. An EUA isn't a full approval. And so you can't do a trial of convalescent plasma without getting an IND. That will take several weeks. So I think just earlier this week, the NIH had said we need to have randomized trials in convalescent plasma, and everyone takes a big breath and says, okay. But then you have to design the trial, you have to submit it to the FDA, you have to get it approved. There's just this sagging time, this delay. So it's overcomable. I don't know if that's a word, but it's my sense of regret about the EUA is that time is lives, and the action has delayed the speed with which we get into generating randomized evidence for convalescent plasma. There was a second half. Remap.
Why haven't people used the adaptive platform design?
The adaptive platform design is new. People haven't used it because it wasn't the way people used to do trials. Randomized clinical trials got going after the Second World War using something called frequentist statistics, and they've gradually built over time, and it got up to the point that the global medical community was filing about 1 million new RCTs per year, but the whole world just got used to doing the same kind of design over and over. And that got hardwired into regulatory authorities. Everything got baked into the system of how we approve drugs, etc. These adaptive platform trials have several features that can make people a little uncomfortable because they're unfamiliar. They use something called Bayesian statistics. It relies much more heavily on relatively complicated computer software. Not everyone is comfortable with that software, so there's a familiarity issue and there's a competency issue. But I can say that adaptive trials have absolutely started to become very popular, especially in response to COVID-19. Indeed, even the recovery trial we talked about in the UK is a form of adaptive platform trial, and several of the NIH initiatives and the initiatives under Operation Warp Speed are also flavors of these adaptive platform trials. So we are totally early adopters, but I think we would say we're very encouraged to see that many other groups are now also getting on board.
And I may just add that part of the reason why these have not taken on is that you also need a digital infrastructure in order to embed all the information that comes from the Bayesian randomization and algorithms. And one of the advantages that we have had here is that we've been able to leverage that digital infrastructure that our IT colleagues have put in place here at UPMC so that we can then enable the performance of these Remap trials.
Yeah, I would, what we've done at UPMC, it totally took a village, and it was a huge part of our clinical staff, folks in the Wolf Center, Oscar and his team, and everyone in the eRecord team, and actually the clinical trialists, those of us sitting in a room designing trials. This thing would have never left the blackboard or never left the whiteboard if it wasn't for the whole enterprise.
Next question, Holly Patterson, Greenville Record Argus. Holly, please go ahead.
Good afternoon. My question is there a, are you seeing cases of repeat positive results in any of the patients that you service? And is there a way that those patients are going to end up in a different trial?
So we do see cases in which someone who has virus detected, and whether or not they either have symptoms and recover or never had symptoms, that if we test them at a later point, we can still detect some virus. That's not particular to the UPMC experience. It's not the common experience, but it's also seen. If you go back to what we talked about what that test does, particularly the viral presence test, it looks for a tiny bit of fragment of virus that's embedded in the cells and usually in the back of your nose, and the test amplifies it up. And you can imagine that you could have completely fought off a virus with or without symptoms and still have some fragments of that virus, perhaps covered in antibodies, not causing you any problem at all, that could be detected weeks, maybe even months later. The real clinical question is, can I get infected, get completely normal, and then get reinfected another time? And we simply don't know much about that yet. Again, we're less than a year into the entire global experience with this particular virus. What we know is that most people who recover don't clinically get sick again. And if you've heard about a report about someone who had the virus, got better, and then had what looked to be a second version of the virus, that person wasn't actually sick the second time. So it doesn't mean that they had no immunity. It may mean that they actually had perfect immunity. Again, this is one of these things that we're learning more and more about it. We often don't enroll people in therapeutic trials that target the virus after, if there's some question about did they ever clear the first time or not. That's more of a pragmatic issue about making sure that we have the right people that we can study and make the right conclusions about.
Yeah, Don said it perfectly. So we have in place that you can't be re-enrolled within 90 days, but it's not because of any particular clairvoyance that we had about COVID-19. It's really a very standard feature in many clinical trials. Your question is a really smart question. It's definitely in the back of people's minds about whether people could become reinfected, but at this point, it's certainly not risen to the fore as any kind of observed major issue at all, but we're still early in understanding this disease.
Well, we've about run out of time, so we have time for one last question that was emailed to us. Now that we have better treatments and UPMC is having such good outcomes, do masks even matter anymore? There's been so much controversy, can't we just stop wearing them?
The masks absolutely matter. Don't misunderstand that. It's the single biggest thing you can do to protect yourself and, more importantly, the people around you who you can't know about how vulnerable they are. Keeping the virus physically either in you if you're carrying it or away from you is the single biggest step. If we can't master this, and it's simple, put it on, wear it right, we run the risk of having all the other broader interventions have to be imposed to help us get control again. I don't think we have to do that again. I think one of the impressive things about the communities we serve, not just Western Pennsylvania, but New York, Maryland, Central Pennsylvania, is how our communities have embraced in general the guidance, and in particular the masking guidance. And we've seen very good experiences. The mask is probably the single biggest reason for that. Don't deny that. It's real. Wear it whenever you can.
Yeah, it's so true. I think the only successful instances we have of successful social interaction and anything that is a facsimile of getting back to normal in the presence of COVID-19 has taken place in situations where people have been very attentive to wearing masks, whether it's our own healthcare workers interacting with a huge number of patients and having a near zero problem with nosocomial infection. That's all because of adoption of masks. And then similarly, in social, whether it's sports gatherings or other businesses, barbershops, the only instances where this has proven to be successful have been when people have still stuck really religiously to what, as Don said, can be irritating but is incredibly important: wearing masks.
Okay, thank you. And that concludes the time we've allotted for today. Thank you all for attending. In about 45 minutes, we will email media attendees a link to download a high-definition footage of the entire press conference, which we encourage you to use in your news reports. Reporters with follow-up questions can contact UPMC Media Relations at upmc.com/media. And please remember to follow @UPMC News on Twitter for updates. Again, thank you for joining us today.